Hello, Stats class. This is lesson 42C, which is how to improve experiments. So I want to remind you of what we've done so far. We've done the simplest kind of experiment only, which is called the completely randomized design, or completely random, either way. And the completely randomized is kind of like an SRS, except it's for experiments instead of samples. So just like with an SRS, we make a list of our subjects, we use randint or maybe table D to choose some of them to be in one group and some of them to be in another group. Or sometimes you draw names of your subjects or possibly treatment names out of a hat. It's actually easier to do the treatments. So just picture doing this in a classroom, and I would have two cards, and I would randomly hand you a card, and you'd look at it and you'd know what treatment you were, and then I'd take it back and shuffle them again and randomly hand another person a card. That could happen. So shuffling cards is another way, and that could be either with a hat or without. You could deal cards into two piles, and that represents the two treatments, something like that. You could do coin flips, although there's a disadvantage to coin flips that I should probably talk about. The problem with coin flips is if I'm flipping a coin, actually this would work with shuffling cards also. If I only have two cards and I deal you a card and then put it back and then deal another person a card, or if I flip a coin for each person to say heads is one treatment and tails is the other, it doesn't guarantee equal groups. And sometimes that might be a problem. And so if you really want equal groups, what you do is you take the cards and you make sure there's enough cards for everybody. So if you have 20 people, you make 10 cards of each treatment and you pass out the cards that way. And then you make sure that you get 10 of each. And with coin flips, what's usually done is you flip coins until you have one group full and then the rest of them go in the second group. And in that case, you'd want to make sure they're well mixed to start with. So you wouldn't want to take like the people as they come in the door. There's some bias that could enter there. Like imagine if all the tardy people ended up in the same group. That's not probably a good idea. So think about things like that. But that's the completely randomized design, is just assign people to groups, nothing else complicated going on. And the problem that often happens when you do that, in fact, I believe we saw this in the warm-up, dealing with the overlap between your treatment groups. So we often see graphs that look like this, where there's some data and maybe an outlier or two, and then we have some more data and maybe mostly gathered in this direction. So the mean is here or so, and the mean of this is slightly lower, but it's not a whole lot lower, and there's a lot of overlap between these two data sets. So just because the mean's a little bit lower, you can't tell if it's because of the treatments, because there's so much overlap going on, and that's because of the confounding variables. Overlapping data like this makes it hard to see the differences between the treatments. And the differences between the treatments is what we're trying to see. So these two graphs show that there's not much difference between the means, which are the little triangles. And so we've got two types of variation here. We've got what I'm going to call good variation and bad variation. Good variation is what we want to see. It's differences between the treatments. We're trying to prove that our treatments make a difference. Bad variation is the differences within each group or within the treatments. Individual differences in our subjects that cause more spread within each group. And those differences are generally caused by confounding variables that are causing some people to have higher whatever our response variable is. So this bad variation is caused by confounding variables. So the solution to that problem, you knew I was going to talk about a solution, right? The solution to that problem is called blocking. And what you do is you block by a potential confounding variable. And I'm going to explain how to do that. So it's actually very similar to a stratified sample, except they don't call it stratified with experiments, they call it blocking. So that's a little bit of a vocab thing. Blocking, what you do is you group similar subjects. So you make groups based on your confounding variable. Let's say you think that exercise is going to make a difference in people's heart rate. So people who exercise often get to be put in one group, and people who don't exercise often go in the other group. And that part is not random. I want to point that out to you. But then you do your randomness, and what you do is you randomize within each group. Randomly assign treatments. Within each block or group. Because what you want to do is you want to make sure that the people who exercise often, for instance, get both treatments. Some of them are in each treatment group, and then the people who don't exercise often, some of them are in each treatment group, so that you balance that confounding variable. You're basically forcing the two groups to be equally assigned to the two treatments instead of just taking it by chance. And then you compare the results within each group. So you can see that this is a lot like stratifying because we're grouping similar subjects with each other, like the salad bar example that I gave you before. And then instead of randomly choosing some from each group and just not using the rest, we're using everybody, but we're going to randomly assign the treatments within each group. And that's what's different about this one. So let's do an example with the pulse rates that we saw in the warm-up. We could, instead of exercisers and not exercises, we could start with your resting heart rates. So everybody takes their heart rate or pulse rate with just sitting, right? And then we group the higher ones in one group and the lower ones in one group. Or maybe we do more groups than that. Maybe we have a medium group. And then we randomly assign sitting or standing within each group, and we do the whole experiment. It's kind of like duplicating the experiment with two different groups of people. So that's what you do if you have a confounding variable that is categorical, then usually you group it into groups. Heart rates are not categorical, they're quantitative, so we could get even better with the heart rate thing. And so let's talk about that. Basically, the smaller your blocks are, the better results you're going to get. So even better would be to group them into, instead of high and low, you could group them into high, medium, and low, or you could group them into high, slightly high, medium, slightly low. That's what, you could do 5 groups, you could do more groups is better. And the best way really is matched pairs. So you would match up your people, because we have 2 treatments, sitting and standing, you'd match them up into groups of 2 that are very similar to each other. So this is a common variation on blocking. And what you do with matched pairs, as you probably could guess, that this is groups of 2, as similar as possible. So what I usually do in class, which I don't think we'll be able to do this year, is I line you up in order by your resting heart rate. And so I would take the low resting heart rates at this end and make everybody stand in a line with the high resting heart rates at this end. And then I take the 2 lowest and pair them up, and the next 2 lowest, and the next 2, and so on, all the way up to the highest 2 get to be in a pair. And then each pair doesn't get the same treatment, they get one of each. So I would randomly assign one to sit and one to stand within that pair. So that is one type of blocking, is to do pairs. In fact, I don't know if you know anybody who's an identical twin, but they get invitations to participate in scientific studies all the time, because one way to do matched pairs medically is to do people with the same DNA, which is identical twins. So those are really useful to scientists, because you can assume that the genetics is not an issue. You could get rid of the confounding variable of genetics by doing that. What's most commonly done with matched pairs is you randomly assign one to stand and one to sit. You do the test, which is the pulse rate thing. You then would usually subtract. It is most common to subtract the 2 values. From each pair, and analyze the differences. And we will learn some more about that analysis later this year. Analyze. Remember, difference is the result of a subtraction. So that's, big blocks is better than no blocks at all. Small blocks like matched pairs is even better. Even better than that. So we're just, how to improve some more. Even better is if each pair is as much alike as possible. Who's most like you in every single way? And the answer is you. You are most like yourself. And so that is called repeated measures. It's a special type of matched pairs where every subject is going to do both treatments. So repeated measures on the same subject, what that does is it says each subject or each person is a block. And does all the treatments, usually with some time in between. Especially if it's a heart rate thing, you'd want to allow time for the heart rate to go back to normal after you did whatever it is you do. So allow time in between. If it's a drug test, they'd probably wait a week or so to get the drug out of your system before you give them the other drug, right? Kind of common sense stuff. There's no randomness yet, right? So if you haven't thought of it yourself yet, I'll tell you that what they do to randomize this is they randomize the order, because maybe it makes a difference whether you sit first or stand first. And again, if we were in class, we would do this, but I'm still thinking about if I can find a way to do it online. I've got to decide by tomorrow, because that's when this lesson is. So if I think of it in time, we'll do it digitally, but I can't think of a good way to do it right now. So anyway, randomize the order, which is first, and then you would do the subtract. If there's 2 treatments, you would subtract. If there's more than 2 treatments, it would be more complicated than that. Oops, I meant to say subtract. I don't know why I wrote subject. Subtract if there's 2 treatments. So to summarize, because these notes are getting a bit long and we want to stop soon, blocking the how and the why, the most important part right here, how and why. How is you use similar groups that are similar within the group, and randomize within each group. Why is to reduce confounding or reduce the bad variation that's caused by confounding. So in other words, if you know that a certain thing is going to make a difference, if you think about the confounding variable in advance, you should use it to block your subjects. And that will make your results more accurate and more easier to read. All right, that's it for these notes, and I will see you in the next video.